histone acetyltransferase today. So as the name suggests, histone acetyltransferase are enzymes, and the main function of them is to acetylate the conserved lysine residues that are present on the histone protein of the chromatin. So let's begin. So first of all, the discovery. The histone, which the scientists have done a lot of experiments, and by 1996 they were able to extract the first HAT enzyme, the enzyme that can add acetyl groups to histones. And one of the first HAT to be discovered was the tetrahymena protein, which was called T55. And tetrahymena is actually a unicellular eukaryotic organism, and which has the ability to store and also to react with certain enzymes. And this P55 was homologous with the yeast protein GCL5, which was known for its activity to assemble the transcription initiation complex. Similarly, the mammalian protein P300 was also being discovered, which was also having some kind of an similar function of the activation of a variety of genes, and this was also found to be an HAT. And after several observations and experiments, the scientists were able to identify the prominent role of histone acetylation and also in the regulation of genomic expression. So histone acetyltransferase can actually be broadly classified into type A and type B according to their subcellular localization. So type A is actually found in the nucleus, and they are responsible for the acetylation of histones in chromatin, and the type B is actually found in the cytoplasm. And what they do is actually they translate the newly translated histones are acetylated, and that facilitates the assembly into the nucleus and thereby forming the nucleosomes. But the classification which we already saw was the old kind of classification. In the modern classification, we use the structural criteria such as the presence of or the absence of chromodomains, bromodomains, and zinc finger. So what chromodomains, bromodomains, and zinc finger means are? Well, actually, chromodomains and bromodomains are epigenetic regulating proteins. So epigenetic regulating proteins can be actually broadly classified into three: readers, writers, and erasers. And chromodomains and bromodomains come under the reader classification. On the other hand, zinc finger is actually a DNA binding domain that are mostly present in transcriptional factors. Now coming to the classification, the first family, transferases. They're actually named for the similarity with these GCL5 enzyme of the yeast, and they have the presence of bromodomain and chromodomain present in them. And chromodomain is having a specific binding affinity towards acetylated lysine. On the other hand, chromodomain is having a binding affinity on mediated lysine. Only in general, GNACs have involved in cellular growth. Like apart from its activity, they are also involved in cell growth. The next classification is the mixed HAT, and they have an acetyl CoA binding motif, and also the presence of zinc finger is observed in them. So they are also involved in control of transcription, cell growth, survival, and also they are part of DNA repair mechanism also. And they are three smaller family members, P300 has, and also another family which is having the presence of TAF250 domain, which is a transcription factor, and another steroid receptor coactivator. Now, as we already discussed, HAT are part of a huge family of protein, and which use the acetyl CoA as substrate. So acetyl coenzyme A, we know it's coming from the oxidation of the pyruvate, and pyruvate we are getting from the glycolysis process of glucose. And the histone acetyltransferase modify the chromatin histone and are the major players of epigenetic modulation of gene transcription program. So what does epigenetic denote is like? They do not have any direct relation with the genetic code, but rather they are into the changes of the structure of DNA and also the associated histone proteins. So this acetylation then allow transcriptional access to DNA. So it's one of the main function of the histone acetyltransferase. And also HAT are not only the protein modifiers and epigenetic factor, but also have critical regulators of cell development and carcinogenesis. So we will be discussing about in the coming up slides. So this is one of the main function of the HAT. So here we can see the histone, and the DNA is tightly wrapped around the histone octamer. And during the transcriptional activation, the hat acts upon the nucleosome. And in the presence of hat, the acetyl group from acetyl coenzyme A is transferred to the N-terminal of the lysine. And thereby, this neutralizes the positive charge of lysine, and this disrupts the interaction between DNA and histone. Now, these acetylated histones are generally associated with euchromatin and transcriptional activation. Like we know that heterochromatin are transcriptionally inactive, so we see this mechanism mostly in euchromatin. So after the presence of we are getting transcriptionally active relaxed chromatin, and as an antagonistic mechanism, we have the HDAC, which is actually histone deacetylase. They act upon this relaxed chromatin and condense it back to the condensed chromatin and leading to transcription repression. So these are some things we already have discussed. And not only the acetylation, there are many other regulatory post-translational modification, just like methylation, phosphorylation, ubiquination, etc. Then one important phenomenal factor of HAT is that individual HAT can acetylate histone in test tube, but most have negligible activity on intact nucleosomes. So this indicates that in the nucleus, HAT almost certainly do not work independently. They require certain kind of multi-protein complex, for example, the SAGA complex or the ADA complex of the yeast or TFTC complex of the human. So far, we have discussed about the histone acetylation. Now we are going to discuss about the non-histone acetylation done by the HAT. So we have discussed like mostly the histones are acetylated. Other than that, the transcriptional factors or the basal transcriptional factors, the transcriptional activators, or the polyamines, all these can be acetylated using HAT. So when the transcriptional factors are acetylated, this is going to influence the promoter activity and the specificity. For example, which is a basal transcription factor, which is usually done like this position the RNA polymer is properly a promoter. So when we are when it is acetylated, it enhances its function. Next important part is the bromodomain. The bromodomain containing protein, we have already discussed, they are known as the readers. So they have been identified as an important type of readers of acetyl lysine. They actually bind to the acetylated lysine residues. For example, the TAF250. This is a family under HAT. This have double bromodomain, and this recognizes the low level of acetylated histone, and thereby it will be increasing the acetylation of these histones. So far, we already discussed about the acetylation done by HAT, and more specifically, we can call this lysine acetyltransferase because it's not just acetylating histones, it's also acetylating the non-histone proteins. So these HATs are not only involved in acetylation of the histone and non-histone protein, but they are also involved in many different diseases such as cancer, inflammatory diseases, and neurological disorders. So here I'm just taking an example of how it is important in the blood cancer scenario. So this is a brief summary of talking about the role of histone acetyltransferase in normal and malignant hematopoiesis. So malignant just define a cancer cell, and hematopoiesis is talking about the premature blood cells. So as far as we have seen analytical activities like histone on histonic effect of HATs in several cells. Now we are going to take it for specific hematopoietic cells. So when they were actually doing the study, they wanted to know how HAT actually affect the cellular events in the normal and malignant hematopoiesis. So they were doing the studies in a mouse model, and they were doing all the approach were in vivo within the organism, so that they will be getting to know the role and the mechanism of the histone acetyltransferases. So when they were actually conducting the study, they were able to see that P300, CBP, all these plays distinct roles in the hematopoiesis. Also, it was thought that TCF is essentially important in the early embryonic development, but later it was found that GCN5, another HAT, is actually part of this. So PCF is actually homologous to the P300. That's why people actually thought it was actually part of the early embryonic development. But later it was found that the HAT GCN5 is actually regulating the early embryonic development in the mammals. Then later, one of the important thing, or the most important theoretical method that actually derived from this is actually the study of bromodomain. So bromodomain can actually bind to the acetylated glycine. So many proteins have a bromodomain. So they were interested to know like whether all bromodomains can recognize acetylated glycine and how specifically they can bind to the acetylated glycine. So initial people were thinking about HAT inhibitors in order to inhibit the HAT which are actually developing cancer cells or something like that. But later it was like the HAT inhibitors is having a huge amount of like concerning level of chemical reactivity. So it is not actually a good method of therapy. So people are not thinking about bromodomain inhibitors, which is more better thinking about looking into the leukemia creating associated problems. The reason for leukemia is actually the chromosomal translocation. They were able to see the HATs like MOZ, P300, and CBP were actually involved in the chromosomal translocation, which is leading to the leukemia. So after the study, people were able to understand that LAT is having some critical roles in the parthenogenesis of hematological malignancies because they are creating cancer. So it is important to specifically we have to just regulate the activity of these LATs. For that, some drugs have to be made which is more specific to the LATs. So nowadays the studies are going on in order to be more specific and some potential drugs have to be created from the same. So through the study, they were just not trying to know what actually HAT is doing in the malignantic hematopoiesis. They also wanted to create new therapeutical methods which can be useful in the future. And so it's throwing light on cancer treatment in the coming era. So as far now we have discussed what actually HAT means, how HAT is divided different types of its families, and how it is doing histone acetylation, and also how it's important in cancer biology also. So that's all from my part. Thank you.